Mitosis is the process by which somatic cells divide. Remember that somatic cells are all the cells in our body that are not gametes, those that are not sperm and eggs. So the process of mitosis has one job. That job is to generate new identical cells. So our cells, depending what they are, for example, skin cells, die regularly. They wear out. They need to be replaced. So the process of mitosis is the process that actually generates those new cells. So mitosis occurs for two reasons. One is growth and development. So as we grow, as organisms grow, they get bigger. And the way that they get bigger is by cell division. So some of it is cell growth. The cells themselves get bigger. But the majority of growth is cell division. Bigger organisms have more cells, and they grow by their cells dividing. The other reason for mitosis is replacement. This is because cells wear out. Cells grow old, cells wear out. So during the normal process of growth, development, and maintenance of an organism, cells die. This is something called apoptosis. And this is sort of the natural cycle for a cell. An organism's cells in their body can sometimes control cell division. So if a cell doesn't reach all those appropriate checkpoints for mitosis or meiosis, the cell will go through apoptosis. It will die because it's not ready or not able to appropriately divide. Or they just wear out. Think about your skin cells. They're constantly bombarded by the outside environment. They die, they slough off. They need to be replaced by mitosis. So down here on the bottom, we have an onion root tip. We saw this in lab a couple of weeks ago. This is an example of growth and development. The root grows down because the end is dividing. So up here, we have older cells. Down here at the bottom, we have brand new cells because the root tip is growing and dividing down. And these cells at the bottom will grow larger and then they'll divide, extending the distance of that root. And then, of course, we have replacement. Replacement also occurs when cells are damaged. So you get a cut. Your cut heals by cell division. So the general process of mitosis starts with a parent cell who is in interphase getting ready for mitosis by performing DNA synthesis. So we're going to copy our DNA in preparation for mitosis. Now, the major events in mitosis are outlined here. First, those chromosomes condense. They get very, very tightly packed. Now, the reason for this is demonstrated by a pot of spaghetti. So if you have a pot of spaghetti and you reach your hands into a pot of spaghetti and you grab some noodles and you pull them apart, grab a wad of noodles in each hand, some of those noodles are going to break because they're not partitioned correctly. They're tangled up, they're twisted, they're fragile. So chromosomes condense so that they don't get tangled, so that they can be sorted correctly and pulled apart during mitosis. So general process here, those chromosomes condense. These little fibers here are called spindle fibers. They attach to our chromosomes and very neatly line the chromosomes up at the center of the cell. This is to allow them to be separated correctly. Then those little spindle fibers pull the chromosomes apart, one to each side. And after that, the nucleus for each new cell forms, cytokinesis occurs, and we end up with two daughter cells. So one of the key points of mitosis is that we produce identical daughter cells. Now, by identical, I mean they are identical to the parent and identical to one another. And also, an important thing about mitosis is that we maintain chromosome number. So if we start with 46 chromosomes in the parental cell, we need to end with 46 chromosomes in the daughter cells. So let's talk about these steps in mitosis. Now remember, we're going to do this in lab too, so I'm not going to go over a lot of details here because you're going to get the details twice. If you are unsure what's happening in any of these steps, a great place to look is always your textbook. Your textbook has fabulous pictures, and I love in the mitosis and meiosis section that they break down each stage. So if you're not 100% sure what's happening, always go back to that book, check out the description in those images because there's a good summary. One thing I want to point out is that for both mitosis and meiosis, you need to know what happens in all of those steps, okay? So if I ask you to identify or tell me what happens in metaphase of mitosis, you need to be able to tell me what happens in metaphase of mitosis or any of the other steps of mitosis or meiosis. So getting ready for mitosis, before mitosis begins, we replicate our chromosomes in the S phase of interphase. Now this produces something called sister chromatids. So replication produces these sister chromatids. So if we start out with our chromosome here and we do replication, remember we're going to pull apart each of our parental strands. So within this chromosome, this singular line that we have, we've got our double helix. Remember, we're going to pull that double helix apart and we're going to produce two identical DNA molecules. Now, in preparation for cell division, those molecules of DNA are connected at the centromere. The centromere is basically the center point of the chromosome. So these two chromosomes are now called sister chromatids. Sister chromatids are identical chromosomes attached at the centromere. They are made by DNA replication. Now, because they are connected at the centromere, this is one chromosome. So we have one chromosome made of two chromatids. So there's lots of similar sounding words here. Each piece of DNA is called a chromosome. Chromosomes have a centromere. When they are still connected at the centromere, those sister chromatids are considered one chromosome. Now, to reinforce this, there is an amoeba sisters video on Blackboard. So, as our cell gets ready for mitosis, those sister chromatids, that single chromosome made up of two identical chromatids connected at the centromere, are going to condense. And they're going to get nice and tightly packed. Now this X shape that we see is probably what most of you are familiar with when you see pictures of chromosomes. This is actually one chromosome made up of two identical sister chromatids connected at the centromere. So as mitosis begins, our chromosomes are in their sister chromatid form. Our cell begins to produce a spindle, which is a little organelle out of which spindle fibers grow. Now those spindle fibers are going to attach to those centromeres, and they are going to pull the sister chromatids to the center of the cell eventually. For now, in preparation of mitosis, chromosomes replicate, producing sister chromatids, which condense into nicely packed chromatids. And then mitosis begins by the formation of that spindle and those fibers that are going to move our chromosomes around. So before we go any further, I want to introduce a couple of terms. First of all, the word diploid. Now, di means two, ploid means chromosomes. What this means, diploid means, is that we have two copies of each type. One from mom, one from dad. Now we indicate that a cell is diploid by using the notation 2n. That means we have two copies of each chromosome, where n is the number of chromosomes, in the cell. So for humans, we have 23 types. So that's n. And in our normal cells, we are diploid. We have two copies of each type. So in our human diploid cell, we indicate that it is 2n equals 46. We have 23 types. Mom and dad each contribute 23 for a total of 46 chromosomes. So in humans, 2n equals 46, our diploid number. All of our cells are going to have two copies of each type of chromosome. Now, that is represented in this cell here by different colors. These blue chromosomes are our two copies. These blue chromosomes, or these two green chromosomes, or these two yellow chromosomes are our homologous chromosomes. Homologous chromosomes are the two copies that come one from each parent. Now, they contain the same genes along the chromosome, but differ in alleles of those genes. So let's say on this blue chromosome here, we have a gene for eye color. So at the top of each blue chromosome here, we might have eye color. Now this dark blue chromosome might code for blue eyes, but that light blue chromosome might code for brown eyes. Okay, so they have the same genes, but they may differ in alleles. And that's because one of them came from mom and one of them came from dad. Your parents are not identical, right? None of them should be. So they should differ at their alleles for many of those genes. Now remember that sister chromatids are those identical copies that are made in DNA replication. So here's our sister chromatids. So what we actually have represented here are homologous blue chromosomes in their sister chromatid form. Now, if we were to count these chromosomes, we would count the number of centromeres. So we have 1, 2, 3, 4, 5, 6. So we have 6 total chromosomes, but we have 2 of each type. Remember, 2 of each type is diploid. So our diploid number for this cell is 2n equals 6 because we have 2 copies of each type for a total of 6 chromosomes. If you have questions on this, please ask. So we will relatively quickly go through the stages of mitosis. And when we get to meiosis, we'll go relatively quickly through those stages. Once we get to meiosis, I will point out some of the differences. I am most worried that you understand the general steps that happen in each of the stages of mitosis and meiosis. And then also that you understand the beginning, the starting point of mitosis, and the characteristics of the cells at the end, and then the differences between mitosis and meiosis. So here we have on the left-hand side interphase. So remember, in interphase, our cell is growing, it's doing its job in the organism. As it gets ready for cell division, it copies its DNA. Once all of those checkpoints are met, once the cell gives the nucleus the go-ahead to start dividing, we leave interphase and enter mitosis. So in interphase, we've got our nuclear membrane intact, protecting that DNA. We've got the rest of our cell and the cytoplasm. Remember I said that we need to make those spindles because that's where the fibers are going to grow that attach to our chromosomes and move them around the cell. In this picture here, we've already undergone the S phase. So we've already replicated our chromosomes. So if we look here, we've got a chromosome here and its copy, it's a little bit longer than that, connected at a centromere. So we have sister chromatids ready for replication. Now the point of making those copies, the point of producing those sister chromatids is so we can allocate one to each new cell. All right, so we've reached our checkpoint. The cell has said, yep, you're good to go. You may enter mitosis. The first phase of mitosis is called prophase. Now in prophase, a couple of things happen. The nuclear membrane begins to break down. Those spindle fibers begin to form. And your chromosomes get nice and tightly packed. So here we have our spaghetti chromosomes. Here we have our nice tightly packed chromosomes. Remember, we don't want them to get tangled, damaged, or broken. As we move into the next phase, metaphase, those spindle fibers are going to grow and are going to attach to our chromosomes. And they're going to attach to each one. And they are going to pull those chromosomes to the center of the cell. This is called the metaphase plate. You might hear me talk about that in lab. This is basically the equator of the cell, and all of the chromosomes line up end to end, all facing the same direction, with their spindle fibers pulling out to the north and south poles of the cell. The reason that they line up at the center of the cell is so that those chromosomes can be evenly distributed. What's going to happen is that these spindle fibers on this side are going to pull down for each chromosome, and these are going to pull up, and you're going to pull apart your sister chromatids. Metaphase is the most obvious phase in mitosis because all of those chromosomes are going to be tightly compact, lined up in essentially a straight line formed by their centromeres along the middle. Once everything is lined up, these spindle fibers begin to shorten, and we start the next phase, which is anaphase. In anaphase, those sister chromatids are pulled apart. You can see that we are no longer attached at our centromeres. We have separated our sister chromatids. Now, at this point, we now have separated chromosomes. Each of our sets of chromosomes is going to become the new nucleus in the next phase. So one full set of chromosomes is pulled to the South Pole. One full set of chromosomes is pulled to the North Pole of our cells, or right to left, whichever direction your cell happens to be drawn. Anaphase happens really quickly. Those spindle fibers shorten, they pull the chromosomes apart. Our last step in mitosis is telophase. In telophase, those chromosomes decondense, and the nucleus, the nuclear envelope, begins to reform. As that is happening, cytokinesis occurs simultaneously. In cytokinesis, the cytoplasm and all of the other organelles in the cell are partitioned, and the cell essentially pinches in. If you're an animal cell, it pinches, it cinches, like if you pull a rucksack or a laundry sack, it cinches. The plasma membrane splits and fully encloses either of the cells. If this were a plant cell, the plant cell wall is actually built up in between. But in either case, as telophase happens, as the nucleus begins to reform, as those chromosomes decondense, the cytoplasm is divided, and we end up with 2 daughter cells. So a quick summary of mitosis. We start with one parent. We produce 2 daughters. Our parent cell is diploid. In this case, our cell was 2n equals 6. 2n meaning we have 2 copies of each type of chromosome. Our daughter cells are also diploid, 2n equals 6. If this was a human cell, we would be 2n equals 46 and 2n equals 46 at the end, because we want to maintain chromosome number. Now remember also that these cells are genetically identical. So the 2 important things, well, 3 important things actually about mitosis, is that we maintain our chromosome number. Those chromosomes are genetically identical to one another and to the parent, and we produce 2 cells at the end. So this slide is really just a written summary of the previous slide. If you'd like to pause here and write this down, you certainly can. These are the things that I want you to know about mitosis. And these are the things that we are going to contrast with meiosis. So sometimes cell division gets out of control. Cancer is the disease that results as a consequence of uncontrolled cell division. Usually what happens in cancer is that a mutation occurs in the DNA that interferes with the cell's ability to regulate cell division. So if you remember in our first video for this chapter, we talked about the cell cycle and checkpoints. Now I just very briefly mentioned checkpoints. Usually what happens is that the regulation of cell division occurs at those checkpoints. If a cell's not ready to go, meaning that all of its DNA is good, it's been proofread, it's checked for damages, then the cell can continue through to the checkpoint. Usually what happens with cancer is that one of those checkpoints becomes disrupted, and so the cell moves through the cell cycle when it's not ready. So cells with errors are copied. Uncontrolled cell division results because of the loss of those regulatory checkpoint elements. So cancer cells are different than normal cells. Our book goes through 3 characteristics of cancer cells. I'm just going to talk about one because I'm trying to keep our lecture length low. You are certainly welcome to check out the section on cancer. I'm going to do it very briefly here. Essentially, cancer cells divide an unlimited amount of time. Normal cells control their cell division by limiting the number of times that a cell can actually divide. So a normal cell has a limited number of divisions, and then it undergoes apoptosis, or controlled cell death. In a cancer cell, because the regulatory checkpoints have essentially been overridden, those cells can divide infinitely, and they divide with errors. They divide with mutations in the DNA. They divide when they're unhealthy. So cancer cells continue to divide and ultimately lead to tumors. So tumors are collections of cells that are continuing to undergo cell division. There are two major types of tumors. Benign tumors are those that can be removed safely, and then basically the damaged cells are completely removed from the body and there's no problem. On the other hand, malignant tumors are the ones that we think about when we're talking about cancer. So breast cancer, colon cancer, things like that. Malignant tumors are usually never fully removed from the body, because in malignant tumors, they shed and spread their cancer cells. They undergo something called metastasis. So metastasis is when basically cells break off from the tumor and spread through the body. Ultimately, cancer kills organisms because the tumor eventually ends up using nutrients and energy that other neighboring tissues and cells would require. So eventually the tumor can essentially block healthy cells and tissues from carrying out their normal functions. So it's not specifically that like the tumor is toxic or the cancer cells themselves kill the individual, but the accumulation of those cells into tumors begins to inhibit normal functioning in an organism. So there are different types of treatment for these cancer cells. We're not going to go into them here. I'm happy to talk about them if you want to set up an appointment with me. Basically, both chemotherapy and radiation work to attempt to stop the uncontrolled cell growth by killing the tumor cells. Ultimately, the cause of cancer is mutation, either mutations that have come about by DNA replication or by environmental factors such as X-rays, smoking, things like that.